and student number is very poor. I did not know why is this type of student. The types of cofactor, last class we discussed about this. The enzymes with its cofactor remove its designed as a apoenzymes. That means these enzymes, again I repeat you, this enzyme actually consists of two parts. One is the apoenzyme part, another is the non-protein. Protein part, that is called apoenzyme part, and non-protein part is called the cofactors. But both these come together, ultimately the enzyme action will be done. So this cofactor will be divided again two parts. One is the prosthetic group, another is the coenzymes. That means actually the cofactor is the part of the enzymes which activate the enzymes and finally to catalytic the reaction. Without the cofactors, enzyme action will not activate. But without coenzymes, enzymes is totally inactive. So enzymes along with the cofactors, this is called the holoenzymes, and this is the actual enzymes. And this enzyme, which basically basic function is to catalyze the reaction. Okay. Next, the substrate. What is substrate? Actually, enzyme, they act on the particular substances. This substance is nothing but a substrate. Actually, some enzymes which act on the some compounds, like say for example amylase, which is act always the carbohydrate like substances. Pepsin, trypsin, erepsin, which is act on the protein like substances, or that is called the protein substrate. In case of lipase, which is act on the lipid substance. And when the enzymes act on this compound, that is called the substrate. And the reactant in the biochemical reaction is termed as a substrate. Suppose glucose or maltose is the substrate, always the enzymes maltase which act on this. So substrate and join they together, just like this type of joining, just like a key factors, or just like some pockets and some definite shapes which is bind with this particular substrate, and these shapes is predefined and particular substance can bind with. That's why the particular substance can bind with the particular enzymes. That means the catalytic reaction will be done. The enzymes, amylase, which is always catalytic the carbohydrate, not the protein, not the lipids. If it is the same pepsin, trypsin and the erepsin, which is act on the proteolytic substances, not lipid, not carbohydrate. Lipase is the same, which is act on the lipids, not in the proteins and the carbohydrate, because their specificity. Enzyme is another most important property is that it is very specific. It's directly a particular substance এর উপরেই কেবল কাজ করতে পারে. They do not react with the any other substance. You are specific, or there's a diploma in the pharmacy courses. Enzyme also same. These are action is the very specific in natures, not act on the all the substrate. They basically bind with the particular substrate and particularly bind them. After that they catalyze them. They form a first substrate enzyme complex, and finally enzyme substrate complex, and finally after catalysis enzyme will be removed and the substrate will be digested or metabolized, or the smaller particle will become. The site of enzyme synthesis, where the enzymes will be site. So we know enzymes are the protein in nature, and all the proteins are synthesized in the ribosome, because ribosome is the factory house for the protein synthesis. And most of the enzymes which are protein in nature, except ribonuclease, so they are or must be synthesized in the ribosome. So that's why enzymes are synthesized in the ribosome, and which are attached to the rough endoplasmic reticulum. And information of the synthesis, actually this information come from the DNA, which type of protein will be synthesized, whatever maybe it is enzymes or not. An amino acid bonded together to form a specific enzyme according to the DNA's code or genetic code, and that type of enzymes or protein will become, and which are acts as a enzymes. But sometimes these enzymes coming from the ribosome which are not active forms. They are sometimes it is also inactive form. But after, say for one example, this insulin, this is also hormone also, but some enzymes, and especially in case of enzymes, there is no modification, but sometimes there is some modification. It is called the post-translational modification. After some modification, these enzymes are active. Another, it is a type of enzymes. One is the extracellular enzymes, another is the intracellular enzyme. Which is called the intracellular enzymes, those enzymes which are produced or active on the inside the cell for the use of the cell itself, that is called the intracellular enzyme. This is found in the cytoplasm, nucleus, mitochondria, and the chloroplast. Especially in the cytoplasm, in case of those enzymes which is act on the glycolysis or in the TCA cycle. I think you do know what is TCA cycle and the glycolysis. This is the first step of the metabolism of the glucose, which is occurred in the cytoplasm, and TCA cycle which is occurred in the mitochondria. Those enzymes which is act on the inside the cell, that is called the intracellular, and those enzymes which is outside the cell, that is called the extracellular enzymes. Say for example, intracellular example, oxidoreductase catalyze the biological oxidation and reduction, enzymes involved in the reduction in the mitochondria. And another is the extracellular, those enzymes which is act outside the cell. That means which will not act on the inside the cell, outside the cell. Say for example, those enzymes which are act on the gastrointestinal tract, like amylase, pepsin, trypsin, or the lipase, which is not in the cell, which is just in the gastrointestinal tract. And these are called the extracellular enzymes which are act in the outside of the cell, that is called the extracellular enzymes. And what are the characteristics of the enzymes? Obviously, I discussed about this. It is a biocatalyst, and main function is the enzymes to speed up or to increase the rate of reaction. Another is that there is a does not affect the nature and the property of the end product. And after the end product, there suppose enzyme E plus S equal to ES, and finally product plus P, E plus P, sorry, E plus S equal to ES plus equal to whatever E plus P, the product. That means after the reaction, the product will become. So say for example, suppose you take starch, and amylase is the enzyme which is act on the starch. After end product is the glucose. So there is no effect on the nature and property of the product, and they are highly specific in their action, and they are, that is the each enzymes can catalyze one kind of substrate. Jodi na hoto tahole they can bind with the or catalyze the any type of substances. I think this is not acceptable, and this is not acceptable in your life also. And small amount of enzymes can accelerate the chemical reactions. A small amount of enzymes can accelerate the chemicals, and there's so much example. In case of a piece of chicken, in case of hydrochloric, keep in the hydrochloric acid, it will be take 2 to 4 days is required to dissolve the chicken. But when the dilute hydrochloric acid along with the piece of chicken and along with the pepsin or proteolytic enzymes, within 2 to 3 hours it will be smaller particle, or it will be digested. So faster the reaction by the agents of enzymes. And enzymes are the sensitive to change in the pH. Obviously, another most important criteria, enzymes are so sensitive in the case of pH. It will be very pH dependent. Proteolyte, some acidic condition, some are act on the basic condition. But the one in case of pancreatic amylase, which is act on the basic pH, which is not on the acidic pH. Another is the temperature. In the temperature, the maximum body temperature, the optimum temperature is the 37 degrees centigrade. That's why our body temperature is 37 degrees centigrade, because our enzyme system act in this temperature. That's why our body temperature, optimum body temperature, 37 degree, because in this temperature, the optimum reaction will be occurred, and the substrate concentration also. This is very, very important, which is the most important factor. Next class I discuss about this, the substrate concentration, how it will be effect on the enzyme action, and when factors affecting the enzyme action, it will be one class at least required, and this question in come and every alternative years. Another turnover numbers is defined as the number of substrate molecules transformed per minute by one enzyme molecules. তার মানে one enzyme molecules turn over the substrate molecule per minute, মানে per 1 minute one enzyme molecule transform the substrate molecule. Say for example catalase, it is the one enzymes, the turnover number is 6 into 106 per minute. Next which is the nomenclature. Obviously this is very important. Every things they are naming, they want all the student having some names, a specific name and their title that will be identify the particular person. Otherwise you cannot be identified. Suppose you are not, no naming system, no name, so there will be difficult in case of identification. So in that case also same. But in earlier days, when the enzymes, there's so many enzymes cannot discovered, at that time there is a no naming, is with non-systemic names. The enzyme is named according to the name of the substrate if the catalysis. Some enzymes were the names in the systemic way, but some are the non-systemic. In earlier days, generally the naming, suppose say for examples, pepsin, trypsin, edapsin. These are the enzymes which act on the proteolytic enzymes. But this type of naming system is called the arbitrary name. যেমন তোমাদের আগেও আমাদের নেমিং সিস্টেমের জন্য মানুষ নাম খুঁজে পেত না, তখন দেখবে ঠাকুর দেবতার নাম রাখতো. This is called the arbitrary name. The pepsin, trypsin, there is no relation between the substrate and the enzymes. This is called arbitrary name. But when that so many enzymes will become, then another problem will occur. Then the naming system will be changed and systemic name will be required. At that time they decided that those enzymes act on the particular substrate, just add the suffix AAC. Say for example maltose, the enzyme is the maltase. Maltose is a substrate and the maltase is the enzymes. Glucose plus glucose. So especially this type of, that means substrate plus AAC equal to enzymes name, and that is called, this type of naming system is called trivial name. The first one is the arbitrary name, second system is the suffix. These are called write down this, these are called trivial name. কি বললাম? These are called trivial name. First one is the arbitrary name, just non-systemic naming system. That at that time yes, few enzymes come and discover, that is called the non-systemic naming system, and that is called the arbitrary যেমন just name নাম, arbitrary name. Then another is the trivial name, which is called the trivial name. Now substrate plus AAC, substrate plus AAC. There are maltase, sucrase, lactose and the lactase, sucrase. So these are called the trivial name. But there is a problem in case of when the catalysis come. Catalase means a common enzyme which catalyzes the reaction, but which is the substrate. This is the problem will become. So how to solve the problem? In case of catalase, there is no specificity, there is no specific substrate, just like a maltase, sucrase or lactase. In the specifically they are act on the particular substrate, but in case of catalase there is a problem arises due to this trivial naming system, because catalase is the enzymes which catalyzes the substrate, not a particular substrate. So at the time in the 1960 a enzyme commission will be come for construct a in systemic naming system of the enzymes. So naming system, say for example, in case of some system, lactose, lactase, maltase, maltase, cellulose, cellulase, lipid, lipase, lipase, starch, amylase, protein, protease. These are the trivial name. But problem hoche in case of, so at that time, how will you overcome this problem? This trivial name, suppose catalase, how will you overcome? At the time, the Enzyme Commission, International Union of Biochemists and Molecular Biologists, they are decided that in the 1964 to formation of a Enzyme Commission or EC, Enzyme Commission, and they construct a naming system, and it was published in the 1964, and that is called EC. After then EC and 4 digit, after that it will be 4 digit number, EC 4 digit, that you can see it. And classification of enzymes, a systematic classification of enzymes has been developed by the International Enzyme Commission or International Enzyme EC Enzyme Commission by the International Union of Biochemistry and Molecular Biologists, IUB, or sometimes some books is written IUMB. And this classification based on the type of the reaction catalyzed by the enzymes. And these are the 6 major classes. We remember that the 6 major classes, what are the 6 major classes, and their class will be and their number will be defined and fixed, not change. Just like there is a chota naam bolbe, you cannot arbitrary, just 6 number according to this you can follow it. First one, major class. And this is classified into a 6 major class. Then it will be classified into the another subclass, subclass, subclass. 4 classification kora hoyeche. There suppose one class, the 6 major class. This is the 6 major class. Its number is very much fixed. Not the ligase is in the first position. Kora cholbe na. Because this number and name are very much fixed. The first one is the enzyme major class. All the enzymes, both the extracellular, intracellular, proteolytic, amylolytic, lipolytic, all enzymes are classified into the 6 major classes. One is the oxidoreductase. First one, even this number and position and the name is fixed. We can understand oxidation, oxidoreductase. What are the reaction? Basically the oxidation or the reduction. Say for example, lactate dehydrogenase, both oxidation and reduction. The lactate dehydrogenase, lactic acid to pyruvic acid. Number 2, transferase, to transfer the group from one group to another, move the group chemical groups. Hexokinase, the example. Then another is a hydrolase. Number 3, their number, be careful, the number and the name be fixed. এই নয় যে ভুলে গেছো, you can change it, that is in the position number 2, you can write down the hydrolase. This is totally wrong. Number 1, oxidoreductase. Number 2, transferase. Number 3, hydrolase. Number 4, lyase, isomerase. Hydrolase, that means to hydrolysis or bond hydrolysis, cleavage with the transfer of the functional group of the in presence of water. Say for example, lysozymes. Then lyase is the non-hydrolytic bond. Non-hydrolytic bond or cleavage, that means cleave, break bond, cleave kore. That is called the fumarase. And the isomerase, intramolecular groups of transferase, that means D to L, L to D, trans to cis. These are called the isomerase enzyme. Triphosphate isomerase, glucose 6-phosphate isomerase. Though their naming is the isomerase, to transfer the D for D to L or L to D, that is called the isomerase. Another is lyase, and this is the just opposite of the lyase, and this is ligase. The ligation মানে joining, synthesis of a new covalent bond. Ligation করা দুটো বন্ডের মধ্যে join জুড়ে দেওয়া, that is called the ligation. And this ligation or lyase is a new bond is formed, that is RNA polymerase, DNA polymerase. Just you can see another slides also. The 6 major classes: oxidoreductase, So alcohol dehydrogenase, and the EC number is the 1111. Actually, this oxidoreductase alcohol dehydrogenase is the first enzymes. Their EC number is the 111, the 4-digit বললাম যে 4-digit numbers. The 4-digit numbers, these first digit signify the major class. I discussed just 6 major classes. That means this ones means it is a major class. 1 means the major class. That means this major class is 1 to 6. Okay, 1 to 6. That means 1 means it is a oxidoreductase type of enzymes. Number 2, it is the subclass. Which class will be changes. And number 3, subclass. And number 4 digit, it is the subclass. Okay, I think you can understand, or any problem. Another, in case of transferase hexokinase, the enzyme EC number 2711. 2 means always transferase. Or you can remember only the position number 1. 2, 3 and 4 digit never did not see. It can seen into the from the internet. Otherwise it can, because this 1 digit, first digit, which is signify 1 to 6, and next digit, second digit 1 to 100, and third digit 1 to 1000, and fourth digit is the 1 to 10,000. So you cannot remember it 10,000 sequentially or 100 sequentially. Very, very tough. Only you can remember it the position number 1. The position number 1 means oxidation, position number 2 means transferase, position number 3 means hydrolase, and position number 4 lyase, position number 5. Position number 5, which enzymes? Isomerase, and position number 6 is the ligase. So these are the isomerase, ligase, lyase, aldolase, the 1-phosphate addition. In case of oxidation, A phase to B, A equal to BH2 and BX. Okay. So these are the, you can, so be careful. I request you to at least you remember it. This is very, very important. What are the classification and nomenclature of the enzymes? Oxidoreductase number 1. That means EC1, that means already oxidorate. EC5, that means isomerase. Any doubt? I think, do you understand? Any doubt? Hello? Hello? Any doubt up to this? Any question?
Sir, arbitrary name, তারপরে আরেকটা নেম গুলো কি বললেন আরেকবার বলে দিন।
ট্রাইভাল নেম। আরবিটারি নেম মিন্স মানে প্রথম যেগুলো বেরিয়েছিল যার কোনো সিস্টেমেটিক নাম ছিল না, যেমন পেপসিন, ট্রিপসিন, ইরেপসিন, দিজ আর আরবিটারি নেম। আফটার দ্যাট some enzymes will come, but there is a problem. And in what basis? Based on the substrate plus AAC. Suppose maltose, this is called the trival name, TRIVAL, trival names. And next is the systemic name, which is done by the enzyme commission. Okay,
sir.
Understand?
Yes, sir.
Any other question?
স্যার ওই সাবস্ট্রেটটা একবার বলে দিন না।
আবার সাবস্ট্রেট করতে গেল?
এই তো বললেন স্যার।
সাবস্ট্রেট তো অনেকেই বললাম। সাবস্ট্রেট মানে হচ্ছে the substances which is bind with the enzymes or react with the enzymes. সাবস্ট্রেট মানে ধরো গ্লুকোজ, মল্টোজটা সাবস্ট্রেট। দিস মল্টোজ AAC, মল্টোজ প্লাস AAC নাম হবে মল্টোজ প্লাস AAC ইকুয়াল টু মল্টেজ।
আচ্ছা,
ক্লিয়ার?
আচ্ছা।
lactose, sucrose. These maltose, sucrose, lactose are the substrate. The nomenclature 1 to 6 is a major class, but another class which will be divided into so many groups that will be not identified, that will be not required. You can go through the internet, and the subclass will be, and list of the name 1 to 10, 1 to 100, 1 to 1000, 1 to 10,000. List will be given, but it will be remembered very tough. But the question will not come. Only you can EC number 1 to 6, that will be identified very easily. Otherwise, you cannot be identified the digit 2, 3, and 4. Okay. Hello.
স্যার
Okay.
অক্সিডো রিডাকটেজ তারপরে ট্রান্স যেগুলো আছে ওইগুলো তো অক্সিডো রিডাকটেজের আবার তো মানে সাব ডিভিশন চলে আসছে এক একটা।
হ্যাঁ, অক্সিডো রিডাকটেজ এটা হচ্ছে মেজর ক্লাস, তাই তো? ভালো করে বোঝার চেষ্টা করো।
6টা মেজর ক্লাস, আবার তার মধ্যে তো সাব ডিভিশন চলে আসছে।
তার মধ্যে আরো 5টা তিনটে সাব ডিভিশন আছে, মানে এর পরেও আরো সেটা এবারে অক্সিডো রিডাকটেজ তো অবভিয়াসলি অক্সিডেশন রিডাকশন করবে, সেই জন্য মেজর ক্লাসের আন্ডারে চলে গেল। কিন্তু তারপরে আরো সেকেন্ড ডিজিট আছে, থার্ড ডিজিট আছে, ফোর্থ ডিজিট আছে, যাদের মধ্যে মানে সাব ক্লাস, সাব ক্লাস, সাব ক্লাস। ওকে, তার মানে অনেকটা ভেতরে অনেকটা রিয়াকশন, সেটা পর্যন্ত তোমার জানার দরকার নেই। মানে একটা যদি মানে কোনো এনজাইমস, তার মানে কোন গ্রুপটাকে ট্রান্সফার করছে, কোনখানে অক্সিডেশন হচ্ছে, কোন গ্রুপের রিডাকশন হচ্ছে, সেটা হচ্ছে সেকেন্ড ডিজিট সিগনিফাই করে। এবার কোন ফাংশনাল গ্রুপ, আরো থার্ড ডিজিট। তারপর কোন মলিকুল কোন ইলেকট্রন ট্রান্সফার করছে, সেটা হচ্ছে ফোর্থ। ওকে, বাট এই যে চারটা ইউ ক্যান রিমেম্বার ইট ভেরি ভেরি কেয়ারফুলি, আজকে থেকে মনে রাখার চেষ্টা করো। ফার্স্ট অক্সিডেজ, সেকেন্ড ট্রান্সফারেজ, থার্ড হাইড্রোলেজ, ফোর্থ লাইজেস, 5 আইসোমারেজ এন্ড লিগাজেস। দিস নাম্বার এন্ড নেম। এরকম নয় যে লিগাজেস উপরে তুলে দিলে ভুলে গেছো, 1 নম্বর দিয়ে দিলাম মানে 6টা লিখে দিলে, তা হবে না কিন্তু। বিকজ দিস পজিশন ইজ ভেরি ইম্পর্টেন্ট। বিকজ ধরো ইসি নাম্বার 311, তার মানে কোন গ্রুপের এনজাইমস? ধরো ইসি 3111, তার মানে এই কোন গ্রুপের এনজাইম? কে বলতে পারে? রেজ ইওর হ্যান্ডস দেখি।
হাইড্রোলেজ।
হাত তুলতে বলেছি, হাইড্রোলেজ বলতে বলেনি। হাত তোলো, রেজ ইওর হ্যান্ডস। আচ্ছা, কতজন হাত তুলেছে দেখি? রেজ ইওর হ্যান্ডস।
কোশ্চেনটা রিপিট করুন স্যার।
হ্যাঁ,
কোশ্চেনটা রিপিট করুন স্যার।
কি বললি?
বললাম কোশ্চেনটা রিপিট করুন স্যার।
কোশ্চেন পিডার হয় না, কোশ্চেন শুনতে হয় কান দিয়ে। ইসি 3111, which class, which is the major class?
বলবো স্যার?
কিন্তু বলেছে। আচ্ছা বলো বলো বলো, সৌরভ সাঁতরা বলো।
হাইড্রোলেজ হবে তো।
হাইড্রোলেজ
3 নম্বর। হ্যাঁ, 3
হাইড্রোলেজ
নম্বর।
সবাই হাইড্রোলেজ বলেছে। সৌরভ সাঁতরা টেলিং দ্যাট ইট ইজ হাইড্রোলেজ। ইট ইজ রাইট অর নট? দিস ইজ রাইট। হাউ মেনি? সুস্মিতা দেওয়ারি ঠিক বলেছে? সুস্মিতা বাড়িতে বসে আছে। অনিমেষ, 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 হ্যালো অনিমেষ, শুনতে পাচ্ছ? ও, এরা মহা ফাঁকিবাজ ছেলেগুলো হয়েছে। চারটে 40। বুঝতে পেরেছো সব? হ্যালো, একবার সবাই ক্যামেরাটা অন করে তো। হ্যালো, একবার সবাই ক্যামেরাটা অন করো। ওপেন ইওর ক্যামেরা।
স্যার আর 50% নিচে অন হবে না।
হ্যাঁ,
আমার 50% নিচে ক্যামেরা অন হয় না, কেন জানি
তোর অত সব ক্রাইটেরিয়া নিয়ে আমাদের চললে হবে? তার হবে না, হবে না, অ্যাটেন্ডেন্স যাবে না। এখন আছে 26, যখনই কোশ্চেন করবো লোকে চলে যায়। লোকের কি প্রবলেম? ক্যামেরাটা অন করে আমি কথা বলছি, ভয় পাওয়ার কিছু নেই। দাঁড়া, রেকর্ডিংটা অফ করে দিই।